Well, I'm representing quite a lot of organizations. As said, I'm leading the Dementia Research Institute in the UK, which brings together from 6 different universities efforts to do something about these disorders. And I have also appointments at the VIB, UCL, and KU Leuven. So just to put it in perspective, I think that dementia is one of the major health problems in the world. And first of all, because of the numbers, and here you see the expected increase over the next 20 or 30 years, which will result in 135 million patients suffering from this problem worldwide. And second, different from other major diseases, it's the only one for which there are no therapies and no hope for the patients at this moment. So when people ask me why is it, well, I think that it's huge underinvestment and a long taboo on these disorders. And so I compared the output or the scientific knowledge we have in different fields of disease. And so here you see the number of papers published per year in the field of AIDS and HIV. And we have at this moment a steady state of about 7,000 papers a year. And here you have the numbers for Alzheimer's disease. So you see that only in 2016 we reached the same level as the AIDS field. And you can also see from the area under the curve that we have about 30% to 40% less knowledge about Alzheimer's disease than about HIV. I think that the crossing of the line is largely explained by the investments in the United States, where there is really a big effort now to tackle these disorders. Everything dwarfs, of course, when you compare it with cancer. So what is Alzheimer's? First, what is dementia? Because both are confused usually. So I show you here instead of a clinical explanation, some painting, some art, it's sometimes nice. So this is William Uttermolen, who painted himself in '96, and he got Alzheimer's disease. And over the years, he continued painting himself, and I think this says much more than many words. Dementia is losing your mind, basically. Dementia is a symptom, and there are many causes of dementia. Alzheimer's disease is, however, the major one. So about 60% of dementia is explained by Alzheimer's disease. But there are several other diseases which have similarities with Alzheimer's disease, and that's why we study all these disorders together in one big institute in the UK. So what's the cause of Alzheimer's disease? Well, the first thing is that it's a proteinopathy. So that's different with viruses or with cancer or with other disorders. Here it's really proteins which cause the havoc. And so Alzheimer's disease is characterized by protein aggregations called neurofibrillary tangles, amyloid beta plaques, which are extracellular tangles, are in the neurons, and then there is also amyloid deposits in the blood vessels. And a second important aspect is that these proteins, also in the other neurodegenerative disorders, spreads over the brain. So they start in specific localizations, and then they go over the whole brain. So it's a kind of internal infection or internal metastasis, but I don't think that these analogies are very helpful. It's a typical signature of these diseases that it spreads progressively over the disease, over the brain. So there are 3 causal genes. We are starting to get better and better understanding of these disorders, and I'm limiting myself to Alzheimer's disease. There are 3 causal genes found for Alzheimer's disease. It means if you have a mutation in that gene, you get the full spectrum of Alzheimer's disease. And so the first gene is the amyloid precursor protein. It's a simple protein, and if you have mutations in it, you get an A beta cleaved out of this protein, which precipitates in the amyloid plaque. So there is a direct link between the morphology of the disease and the genes causing the disease. And so all the mutations are clustered around this amyloid peptide. The second gene, or the second 2 genes basically, are parts of this large enzymatic complex, which is the gamma secretase. There are more than 170 mutations, independent missense mutations, which cause Alzheimer's disease. And the interesting thing is that this is a protease which cleaves this amyloid precursor protein and gives rise to this amyloid peptide. So shorter forms and even longer forms when there are mutations. So one of the big conundrums in the field at this moment is, if mutations in the enzyme and mutations in the substrate invariably give you Alzheimer's disease by increasing amyloid peptide, why is then that the medication which has been developed does not work? And so I think that one of the major insights is that we forget the time factor in this whole problem. And so here you see a typical graph where this is the accumulation of amyloid. And then you see the different other manifestations, tau accumulation, memory dysfunction, clinical dysfunction, which follows. But the important thing is the time factor. And so we know now that this blue arrow takes 20 to 30 years. So people are look completely healthy, have no signs of dementia, but there is already accumulation of these proteins. And so at the moment, we are studying mainly this last phase, which is the clinical phase, which is like we would study cancer at the time that you are completely cachectic and metastatic. There is no cure for that type of disease, or AIDS when you are completely at the end of this end stage. And the field has been largely focused on this end stage because that's the clinic. On the other hand, we have the biochemistry. But as I told you now, this is 20 to 30 years before you are there. And so this is the critical phase. What happens in the brain? What homeostatic mechanisms are you disturbing? What are the cells doing in the brain so that you can keep thinking while these proteins are accumulating? And what finally makes that this homeostatic phase gets disrupted? And so this is just to show you that this is not a linear process, but with feedback loops, and it's a multicellular process. So there are, of course, the neurons and the synapses which degenerate, but you have the glia cells, the oligodendrocytes, the microglia, the vasculature, and probably also the peripheral immune system, which plays a role in this process. And in that sense, we are finally ending where cancer is already a couple of decades, I guess, understanding that these are multifactorial, multicellular processes, and that you need a box with multiple medications to target these disorders in an effective way. So how do we study the cellular phase? And again, it's the genes, stupid. You will hear a lot about environment, etc., because that's easy press. But there is an important twin study, and there are not enough twin studies in our field, but this is really a big one. And this study comes to the conclusion that between 60 and 80% of your risk to get this disorder, to get Alzheimer's disease, is genetic. So it's in your genetic makeup that the risk is present. And this is the sporadic cases. So the 3 mutations I told to you are familial cases where you have a dominant inheritance. This is your chance as a person to get sporadic Alzheimer's disease. So over the years, there are more than 1,000 genetic risk loci now found in sporadic Alzheimer's disease. And this is classical, the Manhattan plots, which most of you know. So you have here the different genetic loci in the human genome, which in combination determine whether you get Alzheimer's disease or not. And so you can look at a high cutoff, which is usually done, so then you get about 20 or 30 genes with high risk. And then you can also take a low cutoff, and then you find thousands and even more genes, or gene loci, which are associated with the disorder. And so then we can use these genetic risks by combining them in what's called a polygenic risk factor. And if you use the major genes, you get a prediction strength, which is indicated by this curve. But if you add even the low risk, the low p-value risk genes, you still add to the predictive value of this model. So if you combine the different genetic factors in these cells or in these humans, you can predict to a good extent your risk for Alzheimer's disease. So that tells us also that mechanistically, in this genetic information, there must be a way to find the mechanisms and to identify new drug targets. So how do we use this genetic information to generate functional hypotheses? I think that's a real question. How and under which conditions will genetic risk manifest? And that's really a very important concept. So it's not equal where you test the function of these genes. It's a bit like when you drive a car and you have your belt on. It's a risk factor if you don't take it, but you will never know that it's a risk factor until you had your car accident. Well, you need to think about these genetic risk factors in the same way. You need to test this function of these genes in a disease-relevant environment. Can we investigate these risk genes in pathological relevant conditions, as I explained? And to what extent, and that's really also a big question, is the mouse a good model to study these polygenic risk scores? Because, of course, the context in which these genes manifest is not only the disease context, but it's also the rest of the human genome which determines how this genetic risk manifests itself. So one thing which we terribly need, I think, in our field is to go away from the classical pathology which we use to describe the disorders and to really use the strength of modern technology to get a full picture of the molecular changes in the brain over time and over space. And this is just to illustrate this again for it's Heiko Braak, basically a pathologist, who described how amyloid distributes over the brain and also how these neurofibrillary changes change over the brain over time. And we need really to document not only in a morphological way these things, but at a molecular level. And so here you see a proof of concept study from my lab where we use spatial transcriptomics and a mouse model with knock-in of the amyloid precursor protein mutations. And what we did is we analyzed this mouse at different stages of disease, so from 3 months to 18 months old. We took a slice which we analyzed with spatial transcriptomics. We took a slice where we did histological analysis, and we added this information, integrated this information in a kind of atlas of what happened there. And so one of the things which interested us very much is what happens around these plaques here that I'll show you in the next slide. So you see here these amyloid plaques spreading over the brain over time, and you see also the cellular response around these plaques which change dramatically. So we developed a bioinformatics tool to measure how close our spatial analysis was with the plaques, and I have no time to explain that, but we have an automatic way to measure the amount of plaques and how close they are to the genes we are investigating. And then we took more than 10,000 different spots in the brain of these mice to see, to analyze the genetic composition. And as you can see, to our delight, these spots cluster according to specific anatomical regions of the brain, and they also cluster according to the time that they are exposed to amyloid plaques. So we think that we have really good data there. And so here just a very short overview of the potency of this type of analysis. So we have here an analysis where we have 2 axes, the amyloid beta load, so the plaque load in the brain, and here the genotype load. And you see at 3 months that we have quite some genes changing according to the amyloid index, so genes down, genes up, and here genes down and genes up. But you see how this really dramatically changes at 18 months, and where we have a whole bunch of genes which change according to amyloid load and genotype. And so by analyzing this type of genes, we can identify novel pathways and understand how the brain reacts basically to these plaques. And here you have an example. So here you have a group of genes which is clearly up early at 3 months, and they're all in the oligodendrocyte, so a cell type which is usually not thought about when you think about Alzheimer's disease. And these oligodendrocytes generate myelin and are very important for the wiring of the brain. So you get a kind of reaction at 3 months, which goes then completely down at 18 months of age, and we can quantify that over the brain regions with the spatial transcriptomic approach, where you see that at 18 months this response is largely gone and is only present still in the hippocampus. We have also an inflammatory response here, which is not high at 3 months, but which becomes extremely high at 18 months, and you can see here in red the brain areas where this response is located. So the problem with mouse studies is that there is a limited orthology of genes, and we estimate that about 30% of mouse genes, 30% of human genes are not reflected by a good ortholog in mouse. And so that's probably one of the reasons why mouse models are limited in value when you try to model complex pathways. And so one of the things I mentioned also in my intervention a little time ago is that what would be extremely helpful to get this genetic information from patients, this polygenic risk score from patients, a tool to study that are iPSC cells, because you derive them from the patients, you have the full genomic information, you can take low risk, high risk, and you can start to do all kinds of experiments. So one of the things I would like to set up in UK and hopefully also in Europe is a huge database of material of IPS, of patient data with genomic data and with clinical data, associated with large cohorts, and to give the scientist the possibility to get access to blood or to cells and to make iPSC cells so that they can study specific genotypes and look what the phenotypes are in the models. And then here, the model which we use a lot at the moment, and it's really a breakthrough, because at the moment we are hesitating about organoids as such, because they are largely embryonic and developmental relevant, but not really relevant for a mature brain. And so we have found a kind of intermediary solution at the moment where we transplant iPS cells in mouse brains and see what happens with these human cells. And here you have a human neuron in such a transplant, so they really reflect nicely human cells. Here you see astroglia, which we transplanted, and they keep this very nice morphology, which you also find in human context. Here, for instance, you see an astroglia which wraps a blood vessel in this mouse brain. And here you see that they react differently to plaques. So these blue things are amyloid plaques, and you here see an astrocyte which heavily reacts, which is hypertrophic. And then here you see an astrocyte which react in an atrophic way. And then there are, of course, also astroglia which do not react. And so it will be fantastically interesting to see how the GWAS genes in this astroglia respond and modulate these responses. So we have also a lot done on human microglia, and this is really cool because they work very well. If you delete the mouse microglia, then the human microglia are sitting in exact niches where the mouse microglia were present. We compared also with single cell analysis whether this mouse microglia reflect human microglia, this IPS-derived microglia compared to primary microglia derived from patients, which we got brain sections from patients who had epileptic seizures and who got operations for that. We got healthy pieces of that brain, isolated the microglia, and we did single cell, and we compared our preparation with each other. Here you see summary. The only important thing here is the in vivo signature of homostatic microglia. Here you see what happens with in vitro generated monocytes and microglia, and you see that the in vivo generated microglia very similar to the primary microglia we isolated. And so here you see an experiment where you see the response of human microglia transplanted in a mouse model for Alzheimer disease, and here you see the mouse response. It's very different, and so I think we are opening a box here for further research. I can skip that slide. Just a mouse microglia give you this, human genes or orthologs, and a human microglia give you this. So what are the most important hurdles in dementia research? For me, compare gene effects at the whole brain level and with single cell resolution. Genomics, transcriptomics, but also proteomics. It's a protein disorder. Allowing unbiased biomarker discovery. Develop in situ methods to access spatial resolution. And then, on the long term, we need better models. In vivo human-mouse hybrids are really revolutionizing the way in the way we do neurodegenerative research. But on the long term, we need 3D derived cultures refined at organ level, brains in a dish, integrated with probes and with measurements in situ. And we need also specific models like blood-brain barrier or glioneuronal unit. This is less important for this conference. So just to say that I think that we need to enhance, to get also the proteomic work into these approaches. I just heard Matthias Mann talking about single cell analysis of proteins in brain, and I think that's really one way to go. And that's here then what I think about organoids. We need to get much more complicated systems which allow us to program the cells in situ, which is possible with these chips, and which allow to let the organoids grow in 3 dimensions, so where the scaffolds give you the way to feed your 3-dimensional structure. And so conclusion, the problem in the field is a narrow portfolio of drugs, so we need to find new drug targets. I think I showed you some way. The clinical endpoints are based on cognition, which is completely wrong. We need upstream and measurable biomarkers. And then we need to get much better data science to pool all the information available. So thank you very much.
Yeah. Thanks for this beautiful talk. In your last remarks about the organoids, what do you think is needed? As you said at the beginning, organoids are good for embryonal development, cell lineage. it might be difficult for degenerative situations. So what do you think would be the angle to?
My approach is a parallel approach. So we are using now this transplantation, which gives us a lot of information on how these iPS cells change in a three-dimensional structure. And I would like to use that type of information as a kind of guidance while we develop these three-dimensional cultures. And so I think the secret there is to provide them with the right substratum, which needs a lot of investigation. I think we should profit while making that right substratum from engineering technologies to bring electrodes into the matrix and bring also small nanochannels into this matrix via which you can transfect your cells. And so you could start to think about induction of your iPS cells in the type of cells you want. And in the long term, the rest I cannot predict. I hope at a certain moment that the cells are very similar to primary cells from brain.
There are people lining up. We start.
I also forgot one thing which is very important for this endeavor. So access to human materials is in the UK really remarkable. There is a brain bank of thousands and thousands of brain frozen, usually with good quality. There are 500,000 samples in the biobank with all genomic material, and they have 10 or 15 cohorts which they follow already for decades, and where you have also access to materials.
You're talking about autopsy.
I'm talking about autopsy.
But during the biochemical phase, this might be
it's less
very
good.
difficult.
So that's why we need these animal models.
Yes.
Thank you very much for that fantastic talk. So my question goes very much with your last comment. Also on your slide, you show the importance of creating disease cohorts and how important it is that they are well characterized. So I wonder whether you could give us some perspective on that, because we have learned how strong bioinformatics has developed, how precision techniques are developed, but I feel that most specimens, even if you have best frozen materials in the UK or so, still the clinical data are sometimes really, let's say, by and large suboptimal. So you're fishing in muddy waters here, and maybe Geneviève and Nikolaus may also kind of comment on it. How do you want to include this kind of building up of cohorts into your consortium? I think it is very important.
It's the Achilles tendon of the whole operation here. There is a big discrepancy in the culture of the clinic and of the research, and it has to do largely with the respect for the privacy of the patient. So, and we all know that if you open the database and you give somebody genetic information, then basically the anonymity is gone. So my approach would be, first of all, there are already very good cohorts in the UK where they follow people from shortly after World War II up to now, and they have consecutive clinical records of these patients, and they have also still access to these patients. So I think with good discussion with these patients, you might get access also to iPS cells and blood, etc. So that would be one of the first ways to go. Second, I think that could be expanded to several other diseases, where you say, look, with Lifetime, we decide to support a cohort in that country for that, a cohort in that country for that, a cohort in that country for that, and we predefine how data will be shared. So everybody who participates in that cohort knows that that will be used for open access under certain ways of secrecy, etc. I think that's feasible. I think the reverse, to get access to huge databases which exist in the clinic, will be, I think it will be Sisyphus labor. So you will roll the stone up and it will turn down again.
We have to look a little bit after time. We have 3 more questions. Nikolaus first.
Yeah, so first of all, thanks for sharing these data and conceptual perspectives with us, and I think you must be aware of it. Your data about Alzheimer's saying that it goes back, maybe 20, 30 years, and it's about finding, this progression early on is exactly the lifetime paradigm. Also thanks for mentioning Matthias Mann with the proteomics. He's actually an active member of Lifetime, so I think there will be more of this in the future. But here's my question. So how far back do you think really goes the divergence of disease to healthy in the Alzheimer familiar cases?
This is really a very funny thing in the field. Everybody says familial Alzheimer's disease is not related to sporadic Alzheimer's disease, and we have been focusing on the wrong problem. Is that the question you asked?
No, the question is about the time scale. So you said
okay.
it starts 20, 30 years earlier, but
Yes.
maybe it's 50 years, right? So
Yes.
How far do you think does it go back?
I think for familial Alzheimer's disease, it's present from birth. So you have the genetic bomb in your genome.
But do you see alterations in the biochemistry of the cells early on, you think?
So the best studies are with the familial Alzheimer's disease cases where
That's
they did
exactly
imaging,
my
etc.
question.
And so that's 20 to 25 years before you get the symptoms of dementia. The official symptoms of dementia that you can see already amyloid plaques and
Because what people see in the organoids built from patients with familiar Alzheimer using controls
Yes.
Where this mutation is cured is very early already alterations. So there are all kinds of complexities to this,
Yes.
but maybe it goes back much earlier than maybe you even think.
I think a parallel approach is certainly indicated, but I would be very hesitant to look. I'm 59, so I think that organoids will deliver in 10 years, and so I don't want to wait 10 years to do further drug development and trying to understand mechanisms. So I will keep my eye open and I will stimulate that in my institute. But I think we need to go parallel. We cannot wait 10 years until the organoids work for doing this type of research.
Yeah. Okay, second last over there.
So your point of that over here, the gamma secretase inhibitors and the other inhibitors are not working is that because you're using them too late, right? So have you tried in your mouse model where you can go into an earlier stage and say, do we have an effect of gamma secretase inhibitors and the other inhibitors in that mouse model to see where is the right time to really do the treatment?
This is the conundrum. So the mouse models are good amyloid models, but the cell reaction on this amyloid is different from what the human cells do. That's why we are developing this. So we didn't do that type of experiment yet with the chimeric mouse model. So in the mouse models, you get a complete cure of the disease, but they are indeed only made to make amyloid. So, so that's a limitation.
Okay, last question.
I would like to comment on the most earliest phenotype on cells, which you see the oligodendrocytes. Is it right? This was also shown for the Huntington's disease also this year from Michael Hayden, and this is also seen by us in spinal cerebellar ataxia type 3 mouse when we screened the proteomics of the mouse all over the disease progression. But my question is, would this also be due to the mouse background? Because it's
these are we don't know.
It's a bit suspicious now. I got a bit suspicious when I saw.
I think I just got excited. Well, I knew this already. So the microglia response is also shared by many. It's like in cancer. You have some concepts which are shared. So all these diseases have abnormal protein accumulations, and they are all killing neurons. And so the reaction of the tissue on this general principles will be somewhat shared. So there will be differences, but there will be shared aspects. And I think it's very exciting to see that these oligodendrocytes are part, I think, of a protective mechanism.
Yeah, we see the early phenotypes on proteomics in 2 months old animals, for example, but don't see the transcriptomics on these animals. So like the proteins are first in our case, spinal cerebellar ataxia type 3.
Okay. Thank you very much again, also for this very extensive discussion.